All right, class. Today we're going to start creating a connection between the probabilities and the alleles and the information that we've been discussing as part of Mendelian genetics and what it is that actually goes on inside your cells. We're going to bring ideas that we've talked about when we discussed the structures and the organelles of cells, and we're going to bring in ideas that we discussed when we talked about things like mitosis, while introducing a variation of that. But what we need to do first is start with a basic idea of what are your alleles. Hopefully when you ask yourself that question, you start thinking, oh, my alleles, those are my traits. You're probably thinking to yourself, oh, those are what's passed on from my parents. Hopefully you're thinking about how you get one allele from your mother, one allele from your father. Perhaps you're thinking about how your alleles interact and how some interactions we call simple dominance, some interactions we call codominance, sex-linked, incomplete dominance, and it is the interaction of those alleles that lead you to your phenotype. But what we need to discuss in this section is what are those alleles actually, and where are they? And so the answer to that question begins with the cell organelle known as the nucleus. Hopefully you're thinking to yourself, okay, well, the nucleus, I know that that is the control center of the cell. I know that the nucleus is where my cell stores information about itself. Perhaps you're thinking about a very important buzzword of DNA. Perhaps you're thinking about when we discussed mitosis and how during the process of mitosis inside the nucleus, chromosomes appear. Maybe all of those things are crossing your mind. And what we want to do is really get to the fact that your alleles are actually sections of your DNA that can be found on your chromosomes. So if we take a closer look at what it actually is that's in your nucleus, what we would find is that here inside the nucleus, we have sections of this organic macromolecule known as DNA. And this DNA, you again hopefully remember, spends the majority of your cell's lifetime uncoiled in this almost like a ball of thread that's been unwound and just thrown in a pile in this structure called chromatin. And now this chromatin, eventually during certain parts of a cell's life, has to sort of wrap itself up. And when it does this, it eventually becomes this structure known as a chromosome. Now, depending on the organism, you'll have different numbers of chromosomes, but these chromosomes, because they're made of this wound up DNA, actually have on them alleles. So when I'm looking inside of a cell, what I'm actually looking at are alleles sitting on your chromosomes. So let's review sort of the connection here. So we start with the idea that sections of your DNA, and your DNA is made of a structure known as a nucleotide. Nucleotides exist in base pairs, and you have billions of base pairs. So you have long sections of DNA. And if we broke that DNA into smaller sections, many of those smaller sections would be what we referred to last unit as alleles. And so sections of your DNA hold your alleles. What that means is that sections of your DNA are what provide you your traits. Your DNA is then organized into bigger sections, which we call chromosomes. Now, mostly we see these chromosomes during the process of cell division, but those chromosomes are still there in certain terms of like one long piece of DNA. And so it's on those chromosomes that we would actually find your alleles. So if we take a look at this diagram right here, what you'll see is that each of these color-coded sections represents a different allele. And what that means is that this one chromosome has multiple alleles on it. And then when your cell goes through the S phase of mitosis of the cell cycle to get itself prepared for mitosis, each of these alleles is duplicated. so that one copy of the allele can go to one offspring cell and one copy of the allele can go to another offspring cell. And that way each new cell has the appropriate alleles. This does add one minor complication to the process of Mendelian genetics. Because many of your alleles are on the same chromosome, some of your alleles are linked. What that means is that before we talked about how if you had an allele for hair color and an allele for eye color, they would, separate and recombine from your parents entirely at random. And in many instances, that is the case. But when we're talking about the physical location on a chromosome, some of these alleles are actually not going to separate and recombine as cleanly as we would like. And that creates something that we call linked genes. And we're going to discuss that a little bit more as we move on. But I need you to understand that because your alleles are on your chromosomes, as an organism, some of your alleles have to be linked because you only have so many chromosomes. So what happens is that during the process of cell division, your chromatin, which is your uncoiled DNA, duplicates itself. And we do this to make sure that each new cell has exactly the same original DNA as the cell that is duplicated. So what happens is, after we get through the S phase, because that's when this happens, the cell goes to G1, it prepares for mitosis, and then it ends up in prophase. And hopefully, one of the things that you remember about cell division is that during prophase, your chromatin coils up into these structures known as chromosomes. And so we are actually able to see these chromosomes, and therefore we can see your DNA in small, discrete units. And what we do is we actually can take a picture of your cell's nucleus when it's like that, and we call this picture a karyotype. And so what you have here is a karyotype for a human being. Okay. Now, human beings have a total of 23 chromosomes, but those chromosomes are in pairs. So human beings actually have 46 chromosomes. And you can see here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, and 46. Hopefully you'll note when you take a look at this karyotype, we always are looking at the chromosomes in pairs, and each chromosome has a number, except for the last two. We talked about this when we discussed sex-linked traits, but these are your sex chromosomes. And hopefully you'll realize because one is significantly smaller than the other, that this is a male. Okay? But there's a lot of information you can gain from karyotypes. Because when we look at these pictures of chromosomes, we know, because scientists have done a lot of research and we have lists that we can access, that on this chromosome, chromosome 5, there are certain alleles. We know from research that we've done and collections of information that we have, that on this chromosome, chromosome 15, there are certain alleles. And so we can actually get a broad sense of whether or not someone has issues by taking a look at their karyotype. For instance, take a look at these two karyotypes. What you'll notice is that the first karyotype, which is our original individual from the last slide, has 46 chromosomes. They have 2 chromosomes at each of the 22 positions, and they have an X and a Y, so this person is a male. What you'll notice of this individual is that they have 2 chromosomes at each of the positions, except for position 21, where they have 3 chromosomes. And you'll notice that 3, We're referring to that as a trisomy. You also should notice that this person is a female. Okay? So what we have here are a male, we have a female with an extra chromosome. Now, typically we think to ourselves, oh, extra is good. Well, in this case, anything other than the exact appropriate amount is not good. Okay? What happens is each of your alleles is a chemical that your body produces. And so if you have an extra chromosome here, what you actually have is multiple alleles on this chromosome that are all producing extra chemicals constantly in almost every cell of your body. And in some cases, it is every cell of your body. That adds up, and it actually alters your allele and can lead to a host of symptoms. And in this particular case, it's a host of symptoms that we refer to commonly as Down syndrome, which is named after a scientist, Robert Downs. So one of the things that we should hopefully learn here is that karyotypes can be very useful to identify broad health problems. And frequently, we actually have a lot of prenatal screening for mothers so that they can actually see the karyotype of their child very early on in the development of the child. And then this allows us to see, allows the mother to see if there are potential issues in their child's future, and then make an appropriate decision for themselves and their family. And that's an important thing for science to provide us that. So it's important that you take away a couple of key facts from this presentation, all right? First is that chromosomes are pieces of cells that contain multiple alleles. You should know that chromosomes are actually made of DNA. You should know that chromosomes are found in the nucleus. You should know that chromosomes start as chromatin, and that chromatin coils up to become chromosomes. And you also should understand that we can actually identify mistakes at the chromosome level and see how they're going to affect multiple traits on a living and functioning organism. And when this happens, when we're actually identifying these mistakes and taking a look at them, we are doing this by looking at what we call a karyotype, which is a picture of a chromosome. Or, I'm sorry, not a chromosome. It's a picture of a cell's nucleus. The only caveat I want to offer there is that picture can only be taken during mitosis. All right. So that's some basics about chromosomes and the first step in our understanding of the connection between passing on your alleles and Mendelian inheritance, and in terms of how those alleles are physically present inside your cells. All right, see you next time.